Today's video is on nucleic acids. Nucleic acids are made up of nucleotides. These are small molecules with a three-part structure. So part number one here is a 5-carbon sugar made up of ribose or deoxy ribose. Ribose is going to go on to form RNA, and deoxy ribose is going to go on to form DNA. It contains a functional group, and also contains a phosphate functional group. It contains a nitrogen-containing base. Okay, so it's going to have a phosphate, nitrogen base, and a 5-carbon sugar, either ribose or deoxy ribose. The nitrogen bases all have carbon and nitrogen atoms linked in rings with functional groups attached to some of the carbon atoms. Okay, so that's going to help it stick together, is what we'll see. Nucleotides fall into one of two categories, either deoxy ribose nucleotides or ribose nucleotides. Again, deoxy ribose is going to form DNA, and ribose is going to go on to form RNA. So, the bases that are for deoxy ribose nucleotides, again, DNA, adenine, guanine, cytosine, and thymine. You definitely need to know these, okay? Know this list. We're going to see that a lot this year. Oftentimes, these will just get abbreviated with their first letter, A, G, C, and T. The bases for ribose nucleotides are very similar, except instead of thymine, we replace that with uracil. Okay, so know these as well. And know that this one, right, is going to be represented by a U instead of a T for thymine. Here we have what a nucleotide looks like if we were to zoom in and look at it on the atomic level. We've got a 5-carbon sugar here. This one is deoxy ribose. So this one's without oxygen. Deoxy is going to be without oxygen. So this is deoxy ribose here. We have a phosphate functional group, and we have a nitrogen-containing base. So notice the ring structures we were talking about just a moment ago. So here's what ATP looks like, right? A moment ago we said it has adenosine triphosphate. So what we're looking at here, here's the triphosphate part of this, right? 3 phosphate groups, okay? So right there's the first one, the second one, third one. Here is your sugar, right? 5-carbon sugar down here. And then here's your nitrogenous base, adenosine. Okay, so that's what it looks like. And it uses these phosphate groups, right, to pay the energy cost. So those will come off of this molecule as it's being used. Okay, so DNA and RNA are the molecules of heredity. and they are nucleic acids. Single nucleotides or monomers may be strung together in long chains by, once again, the type of reaction that we're looking at here would be dehydration synthesis, forming polymers called nucleic acids. So the individual units are called nucleotides. The whole chain is called a nucleic acid. A nucleic acid is an oxygen atom in the phosphate functional group of one nucleotide is covalently bonded to the sugar of the next. So you kind of have this sugar-phosphate backbone is what we end up with, is what you'll hear said over and over when we get more into DNA and RNA, is this sugar-phosphate backbone. The nitrogen base actually moves towards the center of the molecule is what it ends up doing. The polymer of deoxy ribose nucleotides, called DNA, contain millions of nucleotides. Okay, so those are very, very long, and the order is important. DNA is found in the chromosomes of all cells. DNA molecule consists of two strands of nucleotides entwined in the form of a double helix and linked by hydrogen bonds. So I'll attempt to draw this here. Okay, not the best artist, but that's kind of what you got going on. You got a kind of a winding staircase or a winding ladder, like a ladder that you've twisted. Forms a double helix, and it's held together. Right, these bits here in the middle, these are where the nitrogen bases are, and they're actually held to each other by nitrogen bonds. The sequence of nucleotides spells out the genetic information needed to construct the proteins of each organism. So, okay, another function of, put a star next to this, another function of nucleotides is they create proteins right there. They've got the correct genetic information to construct proteins. And so if you have mistakes in your sequence, your genetic sequence, you have mistakes in proteins, and that can lead to some pretty disastrous effects. Single-stranded chains of ribonucleic acid are copied from DNA, and the direct synthesis of proteins comes from within a cell. We are going to see this later on this year a lot of how this actually happens, the DNA to RNA to protein pathway. Okay, so here's just a much better, clearer picture than the one I drew on the previous slide of how DNA is actually linked together. It's actually double-stranded, so here's like one strand, here's the other strand. It's kind of like a ladder that you've twisted, okay? And in the middle, holding the whole thing together, are hydrogen bonds, okay? So they're not covalently bonded. We'll put a star next to this, okay? DNA is actually held together in its helix pattern by hydrogen bonds.